All right, we'll get started here. Thanks for making it to Jake's candidacy. Jake came to us from. This is like a test for the advisor. So as an undergrad, Jake had actually been involved in undergraduate research, which had led to a co-authored publication. So he was ahead of the game when he came to RPI. And I just, it's been a pleasure having Jake with us these past years. I'll give you a few examples. In his first or second year at RPI, Jake put together a presentation as part of outreach efforts to local high schools and middle schools. And I've never seen anything like it. It involved demos on polyvalency. We are now using it every year as part of our outreach with the New Visions program. And so the take home here is anything Jake does, he tends to do well. Over the years in the department, he has received many awards. One of them is an award for his performance as a TA in my own course. And some of those evaluations are to be seen, to be believed. I mean, it's clear he went above and beyond what was needed and was both, in some cases, actually a confidant to students who are now in graduate school and so on. So it was very impressive. Jake has worked hard throughout his Ph.D., but what he has done in the last 6 months has been particularly exceptional. So I'd say more than half of what he's going to discuss today was probably done in the last 6 months. So really very impressive.
We should stay for another year.
Now, I should point out, Jake on the side is, I think, also been paid by Google. And so he's had the misfortune of having a Luddite as an advisor. And so over these past few years, he has slowly first brought me into the 20th century and now into the 21st century. And even today, he sent me some email about Google Hangouts, and I was like, it's too complicated. But anyway, Jake.
Thank you for the and thank you everyone for coming to see my PhD defense. Today, I'd like to talk to you about research that I've done developing multivalent conjugates for the inhibition of a variety of different pathogens: anthrax, influenza, and HIV. So in order to start, I'd like to just make sure everyone understands what multivalency is. Multivalency is simply the interaction of multiple binding ligands on one object with multiple receptors on another object. Now, this kind of interaction occurs ubiquitously throughout nature, and a commonly given example is the binding of a virus to its target cell receptors. Now, the key thing to note about this kind of interaction is that it can often be multiple orders of magnitude stronger than the corresponding monovalent interaction between a single individual binding ligand and its target receptor. So basically all of my research, all of my PhD research has revolved around trying to make use of this concept of multivalency in order to design therapeutics. So what I've drawn here is a cartoon example of a multivalent therapeutic which would bind to the virus, prevent it from binding to the cell, and thus prevent it from infecting the cell. So with that, I'd like to go over an outline for my talk. As I said, I'll be talking about designing inhibitors for 3 different types of pathogens: anthrax, influenza, and HIV. For anthrax, I'll first discuss the intoxication pathway, and for influenza and HIV, the cellular infection pathways. And then in each case, I'll discuss identifying a target in this in the disease pathogenesis, which we think would be good for inhibiting these pathogens. Then, in each case, I'll talk about designing multivalent conjugates, either peptides or proteins, for inhibiting these pathogenic processes. Finally, I'll suggest some future directions which this research should go in and wrap up with the conclusions. So let's start with anthrax. This is a disease caused by infection with the bacteria Bacillus anthracis. This bacteria can form endospores, which can cause infection by inhalation. Because of this, these spores are potential bioterrorism and biowarfare agents. Now, you may know that the disease anthrax is extremely deadly, but what you may not know is that the symptoms and actual death are caused by the secretion of a toxin called anthrax toxin by the bacteria. This is the reason why current treatment with antibiotics is not always that successful, because the antibiotic ciprofloxacin does not prevent the action of this toxin once it's released into your circulation. Therefore, what we really need is an antitoxin to treat anthrax. So let me walk you through the intoxication pathway of anthrax toxin. First, a protein called protective antigen binds to a cell receptor called anthrax toxin receptor, also known as CNG2. And once bound, this protein is cleaved into two fragments, PA20 and PA63. PA63 protein, shown here, can then self-assemble into a heptamer. This is a protein oligomer with 7 units. And once this heptamer is formed on the cell surface, it can bind multiple copies of the other component of the toxin called lethal factor. Once this complex has formed, the entire complex is endocytosed by the cell. And once inside the endosome, the lethal factor can translocate through the pore into the cell cytoplasm, where it causes its cytotoxic effects. Now, previous work in our group had made use of polyglutamic acid as a biocompatible scaffold for creating multivalent conjugates to inhibit this intoxication process. The idea was shown here in this cartoon. Polyglutamic acid scaffolds attached with multiple copies of a peptide, which binds to PA63 heptamer, could outcompete the binding of lethal factor for the PA63 heptamer, and thus prevent lethal factor from entering cells and causing cytotoxicity. Now, as I said, this is a biocompatible scaffold, and it's composed entirely of a polymer of glutamic acid, which is an amino acid, and thus it should also be biodegradable. This is the synthesis scheme, which was developed previously in our research group for synthesizing these inhibitors, in which we create an activated ester on the side chain of the glutamate. And then this activated ester can be attacked by a lysine on the peptide, and in this way we achieve a random copolymer of the conjugate. Now I want to describe for you how these kinds of conjugates are tested. In vitro, these conjugates are tested for their ability to inhibit cytotoxicity to cell cultures. That's what in vitro means. So what is done is multiple serial dilutions of the inhibitor are incubated with a fixed concentration of the lethal toxin. And the ability to prevent cell death is then assayed. And then we can plot this ability to inhibit cell death as a dose-response curve. Now, these curves are useful because they help us to identify a key factor which helps us compare the efficacy of different inhibitors. That is the IC50, or concentration of the inhibitor at which 50% of the toxicity is observed. So you can see for this polyglutamic acid-based inhibitor, the IC50 was in the tens of nanomolar range and was slightly better than a similar polyacrylic acid-based inhibitor. Now, up to this point, when I entered the group, the only method for controlling the arrangement of the ligands on these polyvalent scaffolds was by controlling the feed ratio, the molar ratio of the peptide along and with the PGA scaffold. So in this way, we could control on average, get a statistical average of the percent peptide along the backbone, and by this way calculate the spacing along this scaffold. What we found when comparing scaffolds with different percent attachments of peptide was that there was a point at which the inhibition activity of these inhibitors was maximized, shown by a reduction in the IC50. And we hypothesized that the reason for this increase in the activity was due to the fact that we're matching the spacing of the ligands on our scaffold with the spacing of the binding sites on the PA63 heptamer. This was further corroborated with experiments using liposomes as scaffolds for polyvalent inhibitors. And similarly, we could calculate the average ligand spacing separation on the scaffold by knowing the percent coupling of the peptide to the scaffold, and then could just back calculate what the spacing would be. And again, we see a significant reduction in the IC50, which is good when the spacing matches that of the target. So because of these previous studies in our group, we were inspired to try to design inhibitors that more accurately match our target molecule. That is, the protective antigen heptamer. So let's take a closer look at it. I've highlighted here the binding sites for our peptide ligand in red. And you can see that up to 7 copies of the peptide ligand could bind simultaneously. So we hypothesized that if we could find a scaffold that also had 7-fold symmetry, we would be able to simultaneously bind all of these binding sites and efficiently block this spore and inhibit cytotoxicity. So the scaffold that we found that was appropriate is called beta-cyclodextrin. This is a biocompatible scaffold that's often used as adjuvants, and it's also used as the active ingredient in Febreze, so our inhibitors will smell nice. The only problem with beta-cyclodextrin is that it's relatively small relative to the size of the PA63 heptamer. So we reasoned that we would need to design our inhibitors such that we have linkers to span the distance between our scaffold and the peptide binding sites. This distance needed to span, we calculated as approximately 2 nanometers. And we were thinking that polyethylene glycol-based linkers would be a good choice because they're also biocompatible polymers. However, there had been a debate in the field as to whether use of flexible linkers such as polyethylene glycol would be an effective strategy for the design of multivalent inhibitors. The reason for this is because flexible polymers such as polyethylene glycol can sample a large number of conformations when it's in solution. And it was hypothesized that upon binding to its receptor, the number of conformations available for this flexible polymer to sample would be much reduced, and this loss of conformational entropy would then negate any effects, any enhancements gained by the multivalency of the ligand. So in order to address this issue, my advisor, Dr. Kane, had published a paper in 2010 that analyzed thermodynamics of the interaction between a heterodivalent ligand with a flexible linker, shown here, and a receptor with separate binding sites for each individual ligand. Now, I won't go through all the math, but I will show you the net result, which is that we can predict the dissociation constant of this heterodivalent ligand complex by knowing the dissociation constant of the individual monovalent ligands, and also a term called C effective. This term has units of concentration and can be thought of as a probability with units of concentration, so that's why it's called effective concentration. So what is this term dependent on? Well, you can see that it's related to 2 important features of our multivalent ligand receptor system. That is, D, the distance between the binding sites on the receptor that we're trying to target. And secondly, R RMS, the root mean square distance or length of the flexible linker. This root mean square estimate for linker length more accurately represents the probable length of the linker in solution, because the linker, being flexible, it's not always going to be at its fully extended length when in solution. So knowing this relation between the effective concentration and these terms, we can then take a look at a target receptor, and knowing the distance between the binding sites, say, for example, 2 nanometers, we can chart the effective concentration of the root mean square length of the flexible linker. And what we find is that we maximize this effective concentration term when the root mean square length of the polymer matches the distance on the receptor. This is just a fancy mathematical way to say that we achieve the best binding when the spacing between the ligands matches the spacing between their binding sites on the receptor. So how can we use this for the design of our multivalent inhibitors? Well, if we look at our heptavalent inhibitor design, as I said before, the distance that we're trying to span between our cyclodextrin scaffold and the binding sites for the peptide ligands is approximately 2 nanometers. So if we rechart our effective concentration, this time based on correlating the length of our polyethylene glycol linkers to the root mean square, to their root mean square length, we can see that we again achieve a maximum possible effective concentration. So looking at this chart, we picked a range of different values for our polyethylene glycol linkers to try, as shown here, the first value in each of these series, 2, 3, 6, 8, 11, 12, 15, 21, and 26 ethylene glycol repeats. So then the task was set to synthesize the range of cyclodextrin-based inhibitors using individually one set of these polyethylene glycol linkers. Now this was a complicated synthesis, which was taught to me by a postdoc in our lab, Dhananjay Mondal. I'm not going to go through all of the details of the synthesis, but I will describe the general strategy. And what you should note on the next couple slides is that I've used color to help you see what has been added in each synthesis step. So the first step of the synthesis was to protect secondary hydroxyls on our cyclodextrin scaffold, so that we only attach our PEG linkers at the primary hydroxyls. That is, we only achieve 7 linkers attached. The next step was to add a chemical functionality, in this case an alkyne functionality shown in purple, to the primary hydroxyls. We then used the range of different polyethylene glycol linkers, which were purchased with an azide group, which reacts with the alkyne group shown on the previous slide. But first, before attachment there, I added a chloroacetyl functionality, which is reactive to free thiols. Then I used copper-catalyzed azide-alkyne cycloaddition, which is often called click chemistry for its highly efficient nature, to attach up to 7 copies of the PEG linkers to our cyclodextrin scaffold, shown here. The only step remaining was to attach the PA63 binding peptide ligands. In this case, we used a C-terminal cysteine with a free thiol group to attach to the thiol-reactive chloroacetyl functionality. And in this way, we had our final series of inhibitors that had 7 copies of our peptide binding ligand with a different range of polyethylene glycol linkers. So, just as a reminder, before I show you how these different inhibitors perform, we predicted that the best inhibitors would be for ethylene glycol linkers of approximately 11 or 12 ethylene glycol repeats. Now, I'll overlay the plot of the IC50s of these cyclodextrin inhibitors. These were assayed by a postdoc in our lab, Sunit Srivastava. And you can see that there's a correlation between the effective concentration and the performance of the resulting multivalent inhibitors. In this case, we see no activity for very short linkers. We hypothesize that these are unable to span the entire heptamer, and upon achieving more appropriate spacing with our linkers in this range of sizes, we were able to see the most effective inhibition activity of our inhibitors. Just to compare the performance of the cyclodextrin-based inhibitor with 11 ethylene glycol repeat linkers to the performance of the monovalent peptide at inhibiting the action of anthrax toxin, we saw a difference of 5 orders of magnitude, which is approximately 100,000%, or 100,000 times difference. So this is really good.
Yeah. On the previous slide, how do you know that all the 7 locations are functionalized like this?
So I've done NMR to confirm the yield and of each product, and we have used a range of different chromatography, like flash chromatography techniques to purify, which I kind of stick to.
So all 7 you can attach the ethylene glycol and then functionalize with peptide.
Yeah. Basically, you just added a large excess of peptide to the reaction so that this would help drive the attachment of 7 copies of peptide.
And then does that, the group, the functional group there, does that change the distance slightly? On the next slide.
I mean, it will add a small amount, but the thing is, there's a fairly flat plateau here at the top, so it's not exact.
Okay.
It's not angstrom like exact. So to summarize this part of my talk, I synthesize rationally designed multivalent anthrax toxin inhibitors that match the geometry of the PA63 heptamer and that have improved inhibition efficacy compared to the non-matched designs. And also, in doing so, the use of flexible linkers in multivalent conjugate design is valued. So now I'd like to move on to talking about designing inhibitors against influenza. Now, before I start that,
before you move on, could you just spend a minute to tell us how far you think that is from the clinic? So.
Okay. Sure. So I have done other syntheses of longer polymer base. So if I take you back to the very first slide where I showed the polyglutamic acid based inhibitors, shown here. So I've also done a lot of work with these kinds of inhibitors, in which case we were trying to develop them for preclinical studies. So I scaled up the synthesis here, and they were tested in vivo. Now these are much larger than the cyclodextrin inhibitors, but we did see, I have some slides at the end, maybe once I've gone through, I can come back to your question and address it, because I have some slides at the end that will show the performance of these polyglutamic acid-based inhibitors in vivo. And that will give you an idea. You can compare the performance of these in vivo to the performance of the cyclodextrin. Is that all right?
Yeah.
Okay. So, as I said, I wanted to discuss some challenges for developing or for treating viral pathogens before I get started on the background for influenza. These are that there are often multiple different serotypes or strains of viruses out in the wild. And this makes it difficult to design an inhibitor that will, or a treatment that will treat all kinds of viruses. Secondly, viruses are known to mutate, and the progeny can then acquire resistance. This is due to short generation time of viruses, and the fact that some viruses may also have error-prone gene transcription machinery. For example, HIV's reverse transcriptase, making the acquisition of mutations by the viral progeny more likely. Third, drugs and the immune system both provide strong selection pressure for natural selection of resistant strains of viruses. So because of these reasons, we reasoned that identifying conserved targets may reduce the rate of the development of resistant strains of viruses and would be more amenable to prophylactic therapeutic strategies. So now let's take a look at the structure of influenza virus A. These are typically spherical virions of approximately 80 to 120 nanometers in diameter, and they're enveloped viruses, meaning they have a lipid bilayer membrane, which is often taken from the host cell as it's budding off in the infection pathway. Another key feature of influenza is that it's covered in these spikes. They're glycoproteins, hemagglutinin and neuraminidase, which the hemagglutinin occurs as a trimer and the neuraminidase occurs as a tetramer. And there's approximately 300 hemagglutinin spikes and 50 neuraminidase spikes on average per virion. These two proteins, hemagglutinin H and neuraminidase N, determine the viral serotype which you hear about in the news, for example, H1N1. There are 16 different hemagglutinin types and 9 different neuraminidase types that are known, but only H1, H2, and H3 are known to be able to transmit efficiently between humans. H5 is also an avian strain that is known to be able to infect humans but not transmit efficiently. Now, I'd like to take a closer look at the hemagglutinin spike and its role in the infection pathway of influenza. You can see from the comparison of the hemagglutinin trimer and the monomer that the trimer has these two domains, a globular head domain and a stem domain. This is where the attachment is to the viral membrane down at the bottom. You can see up here the receptor binding site. This is where the hemagglutinin spikes attach to the cell surfaces. Now, if we look at the monomer, you can see that highlighted are several antigenic sites, and these are all concentrated on the globular head domain of the hemagglutinin protein. These antigenic sites are known to be extremely structurally plastic. That is, they can tolerate a range of mutations that allow the hemagglutinin protein to escape recognition by the adaptive immune system, that is, escape recognition by neutralizing antibodies, but still perform its receptor binding function. I'll just also point out the existence of a fusion peptide down on the stem. This fusion peptide has the role of inserting into the cell membrane and drawing the viral and cellular membranes together to allow the virus to physically enter the cell after it's attached via the receptor binding sites. Now, as I mentioned, the primary location of antigenic sites for the hemagglutinin spike are all on this globular head. And this is shown by these antibody structures and attaching to the hemagglutinin head. These antibodies are only specific to, are only able to recognize specific strains of the viruses that express specifically this version of the hemagglutinin spike. However, about 5 years ago, in 2009, a lot of excitement was generated in the influenza research community because antibodies were identified called broadly neutralizing antibodies that were found to bind not to the hemagglutinin head, but to the hemagglutinin stalk. And not only did they bind to a broad range of the hemagglutinin subtypes, almost all of group 1, they were able to neutralize these hemagglutinin subtypes. And the mechanism for this was shown to be that these antibodies stabilize the formation of the fusion peptide in a conformation that doesn't allow it to insert into the cellular membrane and fuse with the cell. So these kinds of antibodies, these broadly neutralizing antibodies, still allow for the virus to attach to the cell, but they don't allow for the virus to enter the cell. So while this inspired a lot of research in the influenza community for trying to design vaccines that can more adequately train our immune system to create antibodies such as these that are broadly neutralizing, our collaborators at the University of Washington decided to take matters into their own hands, and they designed inhibitors that bind to the hemagglutinin in the same location based on their expertise of computational modeling of proteins. I'll skip some of the details, but what you can see is that from these crystal structures, two of their inhibitor designs bind to the same location on the hemagglutinin stem as the broadly neutralizing antibody does. And not only that, but they bind in a way that was very close to their predicted method or structure upon binding. And they're shown here. Also, their inhibitors were able to bind to the broad range of hemagglutinin subtypes, similar to the way that the broadly neutralizing antibody was, as demonstrated by these green check marks. So when we saw this, we thought, wow, that's really impressive, but we can probably make it better. By designing divalent inhibitors, or by taking advantage of multivalency, because the hemagglutinin spike, again shown here in rotated structure. So this is the crystal structure of the hemagglutinin stalk with the designed binders in complex. So when I looked at this crystal structure, I was able to estimate that the distance between binding sites on a single hemagglutinin trimer is approximately 6 nanometers. Then I was able to take a look again at the PEG scaffolds that are available commercially and decide on a range of 3 different PEG linkers which I could use to create homodivalent conjugates that would span 2 of the binding sites rather than just attached to 1. Here's the synthesis scheme for these homodivalent PEG hemagglutinin binding protein conjugates. The PEG linkers featured a Marlene functional group, which is reactive to free thiols, and thus when combining proteins with a cysteine that have a free thiol, we're able to attach proteins divalently on the PEG scaffold. I show the results of the reaction, or I show the reaction products of this conjugation reaction on this STS page gel. You can see that the appearance of bands corresponding to our divalent products is significant and at the expected molecular weight for the corresponding products. I was then able to purify these products by first reducing out the side product, disulfide bonded side product, and then using SEC to purify these reaction products. These were then tested in a microneutralization assay by a postdoc in our lab, Mark. This is an ELISA-like assay. Shown here is a photograph of the plates that were used. What was done is the divalent inhibitors were incubated with the virus for an hour before exposure to cells, and the ability to inhibit the infection of the cells is plotted here again in a dose response curve. These curves were then used to calculate the IC50s of these divalent inhibitors, which are shown here. And we actually had a range of different hemagglutinin binding proteins, which Baker Lab had provided us with. So I was able to synthesize 3 different homodivalent series of PEG conjugates. You can see from this plot that, contrary to what we predicted, we actually got our lowest IC50s with the shortest PEG linkers. And we're still trying to work out why exactly this might be. But another thing to consider is the fact that, as I mentioned, the entire virion is covered in multiple hemagglutinin spikes. So it might be that maybe these shorter linkers more accurately span the distance between spikes rather than spanning the binding sites on a single spike. But further investigation into this. I've plotted, or I've summarize the results of this plot here in numerical form, so you can see that the IC50 of our divalent conjugates were between 3 and 40 times improved relative to the monomer, the unconjugated monomer protein. So this is pretty good, but in parallel, I was also synthesizing polyvalent versions by attaching the hemagglutinin binding proteins to either PGA scaffolds, as I showed in the beginning, using Malamid functional group as the intermediate, or another scaffold that's biocompatible, a polysaccharide called hyaluronic acid. And the synthesis schemes are shown here. Again, first malamids were attached, and then these malamids are reactive to the free thiols and hemagglutinin binding proteins. I was able to characterize the amount of malamid that was added to each scaffold by NMR, as shown here, and then purify out these scaffolds and mark again tested them. And you can see that, so this column again corresponds to the hemagglutinin binding monomer, which was quenched so that the thiol did not form disulfide bonds, relative to our polyvalent conjugates. As you can see, our PGA-based conjugates did not improve significantly, or did not change the efficacy of the hemagglutinin binding protein significantly, but our hyaluronic acid-based inhibitors did achieve a similar level of enhancement as we did with the divalent conjugates before, of about 30-fold enhancement. So this is pretty good, and in summary, I've synthesized multivalent conjugates that improve the inhibition in vitro of influenza infection up to 40-fold relative to the original protein monomer that was designed by the Baker Lab. And continued work is needed to improve the purity of the divalent conjugates, and further characterization of the polyvalent conjugates is needed. Now I'd like to transition to talking about the design of multivalent conjugates that we hope will be able to inhibit the infection of HIV. So this is a cartoon showing the first part of the infection pathway for HIV. As you may know, HIV specifically infects cells of the immune system. That is because the glycoproteins on the surface of HIV bind specifically to molecules that are on the surface of cells from the immune system. These are CD4 molecule and either one of two co-receptors, CCR5 or CXCR5. These receptors are utilized by the immune system for signaling between different cells of the immune system. And HIV exploits these molecules for gaining entrance into the cell. Now, we reasoned that we might be able to design inhibitors for HIV by targeting the CCR5 receptor, shown here, because this receptor is involved more in the primary infection with HIV. And also, individuals that feature a CCR5 mutation, shown here compared to the normal phenotype, are resistant to infection with HIV. And otherwise, they're normal and healthy. So we reason that because of this, we can probably target this receptor and not cause any unwanted side effects. So this was something that we've been thinking about doing for a while, but very recently, a new type of pathogen was targeted that also exploits the CCR5 receptor. This is a leukotoxin excreted by the pathogen Staph aureus. This paper was published just about a year ago, and they showed that this leukotoxin, ED, only kills CCR5 positive cells. Furthermore, they could show inhibition of the cytotoxicity of this leukotoxin by using CCR5 binding antibodies. And they used a range of antibodies to show that they could then identify a probable location for the binding of this leukotoxin to CCR5 receptor. That is, that it's probably ECL2 due to the inhibition of this antibody 45531, which binds specifically at this location on the receptor. Now, an important thing to note is that both leukotoxin components, LeuK-E and LeuK-D, were required for cytotoxicity to cells. That is, neither one component on its own was cytotoxic. Secondly, it was shown that only LeuK-E binds to CCR5, not LeuK-D. So when we saw this, we thought, okay, we've identified a binder to CCR5 that is non-toxic on its own. So maybe we can take advantage of this binding capability of LeuK-E to design multivalent inhibitors of HIV. Now, here's a look at the structure of LeuK-E. And you can see that the rim domain, which is responsible for receptor binding, is down at the bottom. And both termini of the protein are on the other side of the protein. So we reasoned that we could easily use these termini for attachment to a polyvalent scaffold. I used a synthesis scheme based on hyaluronic acid modification and UGA modification, similar to what I described before, attachment of Marlene, followed by incubation with the free thiolate protein. And I just want to mention that the cloning work to prepare plasmids for these leukotoxins was initially done by Dr. Manish in our lab. And then I did all their transformation, culture, and protein purification work. As you can see here, this is these are fractions from immobilized metal affinity columns. And you can see the protein is in the expected molecular weight. And after SEC purification, after the IMAC purification, I got a very pure protein. I was then able to attach this polyvalently. Again, I'm showing you the reaction products on this gel with polyglucamic acid as a scaffold. And you can see that a range of multiple different species of polyvalently conjugated species were created. I then purified these again by SEC, of which the curves are shown here. The black curve shows the trace for the monomer by itself, and then the red and blue curves are the trace for polyvalent conjugates. In which case, after taking these fractions corresponding only to the larger conjugates, I was able to remove the unconjugated protein. These inhibitors were then tested for their ability to inhibit the binding of CCR5 binding antibodies in vitro. And this assay was carried out by Mark. So if we look here, there's 3 different CCR5 binding antibodies. 45531, which I pointed out before from the initial paper on the 2D7 and 3A9 are also well-known CCR5 binding antibodies. A control antibody, which doesn't bind to CCR5, is shown here. And CCR5 negative cells are shown here, also with the control antibody. You can see that for each of these antibodies on CCR5 positive cells, our polyvalent hyaluronic acid conjugates showed inhibition, the ability to inhibit the binding of these antibodies in vitro. This column corresponds to a fusion protein, which was meant to mimic the binding of the HIV GP120 protein to CCR5. And we didn't see inhibition in this case. But because these antibodies shown here are each able to inhibit HIV infection on their own, we still think that these conjugates might be able to inhibit HIV infection, which we'll still have to test. Another thing that we're looking to test is to improve the ability to detect the binding of these conjugates that I made to their target cells. One way to do this might be to use GFP fusion of LeuK-E. As shown in the original paper, they were able to detect GFP they were able to detect the binding of GFP fusions by the GFP signal to CCR5 and inhibit that with 45531 antibody. So again, cloning work was done by Manish, and I created a lot of GFP leaky fusion, as shown here, and again purified by IMAC and SEC. So I'll need to synthesize these conjugates based on GFP leaky and test them further. So in summary, this polyvalent conjugates were synthesized that inhibit the binding of anti-CCR5 antibodies. And because these antibodies are themselves able to inhibit HIV infection, we hypothesized that our polyvalent conjugates might also be able to inhibit HIV infection. But further synthesis and testing of inhibitors based on GFP leaky conjugates is required. So I'd like to just mention a few future directions that this research could go in. One is that we might be able to use an alternate ligand type, which is, so up to this point I've talked entirely about peptide ligands and protein ligands attached polyvalent. But another ligand type is oligonucleotides. These are often called aptamers for their ability to bind to targets of almost any target, which you can identify by doing what is called biopanning or screening. Oligonucleotide aptamers also have the advantage that they often can be identified with low nanomolar or picomolar affinity for their targets. And also, they might be less immunogenic, that is less likely to cause an immune response than peptide or protein ligands. So, because of this, I have screened RNA aptamers against one target, Anthrax toxin receptor 2, and other members of our lab, Manish in particular, has screened for RNA and DNA aptamers that bind to CCR5. In both cases, what we're doing here is I'm trying to identify ligands that bind not just to a new target, or not just identify new ligands, but also ligands or receptors that are cell receptors, and thus would avoid the problems of viral pathogens that I had mentioned before. In preliminary work, I also showed the ability to enhance the affinity of polyvalent conjugates of aptamers for their targets by using a aptamer that was published in the literature called SGCAC. So here's a gel showing a heat map of the resulting of the reaction products of these violated SGCAC aptamers. This is a similar synthesis scheme where I use malamides on a polyvalent glutamic acid scaffold and incubated with the methylated aptamers. You can see the reaction products here. Upon purifying these reaction products from lanes 3 and 5, we were able to show enhanced binding to the target cells, which are leukocytes, relative to the monovalent SGC-8C aptamer shown in blue here. So this is promising. We now know that once we identify aptamers from our screens, which is still ongoing, we're doing some analysis of the results, we should be able to create polyvalent conjugates based on these ligands that are also successful inhibitors. Another future direction I'd just like to mention is bioengineered scaffolds may enable us to improve control over the multivalent architecture in a similar fashion to the way that I had designed the cyclodextrin-based scaffolds for the anthrax toxin. But in this case, we'd be able to recombinantly produce a scaffold based on a repeating sequence of a flexible series of amino acids with designed functionality at designed intervals on the scaffold. The production of scaffolds similar to this has already been started by a second-year grad student in our lab, Chad Garde. And so we're confident that we might be able to use scaffolds like this in the future to more accurately match the spacing of ligands on our polyvalent inhibitor designs. So with that, I'd like to conclude by saying that I was able to enhance the inhibitory efficacy of peptide and protein ligands against anthrax intoxication or influenza infection by multivalent conjugation. I was also able to enhance the binding inhibition of anti-CCR5 antibodies by multivalent conjugation of a recently identified protein ligand against CCR5. This could be a promising strategy for HIV infection inhibition. For the anthrax toxin work, I was also able to rationally design inhibitors based on the geometry of the target protein oligomer. And also I just want to emphasize that exploiting conserved targets may help to reduce the emergence of drug-resistant pathogens. Therefore, we're continuing efforts to identify ligands against the anthrax toxin receptor, the conserved stock of the influenza A hemagglutinin protein, and the HIV co-receptor CCR5. Finally, oligonucleotide aptamer ligands may be able to enhance the therapeutic potential of polyvalent conjugates such as these, and bioengineered protein polymers may enable more precise control over the multivalent architecture. With that, I'd like to acknowledge the support of my Ph.D. advisor, Dr. Ravi. It's been really great working in the lab, and I've learned a lot over the past 6 years. So I'd also like to thank my Ph.D. committee. You all provided very valuable feedback during my candidacy, and it's been a pleasure just getting to know you over the past 6 years. I'd like to thank all the past and present members of the Kane lab, especially the ones that I had highlighted already. Mark for testing a lot of my recent inhibitors, Benajay for teaching me a lot of chemical synthesis, Sunit here for testing a lot of my anthrax toxin inhibitors, Mohan for helping with design of figures and stuff like that. I'd like to thank the RPI staff, Lee Velarde in particular. She's been very helpful. Thank you for coming. I'd also like to thank my family. They've been very supportive of me during my PhD, and especially all of my friends, especially my friends from home and from UConn, who have made several trips up to visit me in the past several years, and without whom I probably would have graduated 2 years ago. I'd also like to acknowledge funding by a fellowship that I was awarded upon first arriving at RPI and NIH. So with that, I'd like to take any questions.
Do we have time for questions from the general audience?
I have two questions. Sure. So, with regards to the influenza inhibiting peptide, so can you tell me how many cysteines are present in the peptide? And if there's more than one, is there any way to control which cysteine is being bound? Or?
No, there was only one cysteine, which was added by the Baker lab to their plasmid design. And these cysteines were, they were the residues of the original. Basically what our collaborators did was they picked residues on the original hemagglutinin binding proteins that they knew to be towards the back end, that is on the opposite face of the protein from the side that binds specifically to the hemagglutinin, thus hopefully helping to ensure that our conjugates don't interfere with the binding.
Second question, you say you use hyaluronic acid as a scaffold in scenarios. I'm just curious if you looked at very narrow average molecular weight.
Now, the hyaluronic acid that I had used was purchased from Sigma, and it's extremely polydispersed and extremely large. I think the starting scaffold molecular weight is about 1.5 megadalton. So I didn't show gels of those reactions because the conjugates are so large they don't even enter in the gel. So
you show me the cycle that's yeah. If you want to make just one reactive site to find your peptide or mediation and block the methanol, other side is blocked. Why do you use non-blocking everything is fine to peptide? In a cycle that's free, you can find three, 1 to 3 peptides you can bind.
I'm not sure I understand the question.
Instead of methanol blocking,
Uh,
yeah, you just
so maybe you're confused. This
kind of bind the
He's saying instead of, so you're protecting the secondary hydroxyl. You're just using the primary hydroxyl to attach the PEG and the peptide.
Right.
He's saying, what if you attach them?
Okay, so you would like to create an inhibitor that has maybe 21 different regulators. That was a strategy we didn't pursue, but I don't have a good depiction of the cyclodextrin. Maybe here. The secondary hydroxyls actually point more inward towards the center of the cyclodextrin. The primary hydroxyls more outward. I'm not sure that conjugation here, I mean, I'm not even sure we'd be able to get conjugation to all of the secondary hydroxyls because it would end up getting very sterically hindered. The attachment of these of multiple PEG linkers to all of these sites on the same molecule would, I'm not sure we'd even be able to achieve that. Does that answer your question?
It was actually tough to even get 7, which is why we had to use like chemistry with the idea that the yields would be high. Because you're essentially coupling two macromolecules.
you expected the negativity effectivity. Negativity effectivity also if you use the same one ligand. If you use the same one ligand, then you will expect the negativity bind. That's it. You can tell.
Okay.
It's saying that whether they will negatively impact you or not.
It might negatively impact the binding just because, as I said, the extra linkers might actually end up getting in the way.
I had a question about your influenza work. When you did the dimer, you calculated the distance between the points. You said it was 6 nanomolar. And then you showed that your most effective one was 3 nanometers. But did you account for the length of your Hep body or your binding domain in the total separation between your two binding sites?
You mean the actual size of the hemoglobin binding protein itself?
No, I'm talking about, because you conjugated 2 protein ligands, the ends of this linker. So did you account for the extension of the entire divalent body when you're calculating the optimal distance between your 2 protein ligands?
So what I did was, as I had mentioned to Brady, the reactive sites were on the back ends of each of these hemoglobin binding proteins. So I estimated this kind of a rough estimate of the separation distance, which I came to this calculation by basically imagining a circumscribed circle around this complex at this cross-sectional plane, which would be exactly at each of the thiol groups there. So then you could calculate the arc length and it was approximately 6.
Of dial groups on the peptide ligand, the protein ligands that are bound to the.
Yeah. Are there any other questions?
Very nice talk, Jacob. I was just wondering if you have to extend this to designing multivalent inhibitors which were not the same or uniformly spaced. Like, so if you have attached different ligands at different spacings, how would you what are your thoughts on that?
Well, actually, the most of the kind of previous generation of polyvalent conjugates we didn't have control or good control over the spacing. So we assumed kind of a spacing which was basically a statistical average. But there could have been attachment of two very close together followed by a long space where there was no attachment. But I think that if you had a target, for example, or maybe I have a slide. I don't think I have a good slide to show you, but for example, the CCR5 receptor, we've had the idea that we might be able to target multiple different of the extracellular loops on that protein. And in that case, we can think about maybe designing an inhibitor where we have two ligands very close together, one for each of the loops, and then we'd like to create a polyvalent inhibitor of these two ligands. So we would then probably have increased spacing between the next set of two CCR5 ligands. So I think depending on, again, your target protein, then you might be able to, or you might want to design a system.
And one of the features was that you showed that you can design a protein scaffold, and specifically, so if I understand it correctly, you're actually, in the design of the protein scaffold, you're including your ligand, right? I mean, if it's a peptide ligand, for instance, you're expressing everything.
Actually, no. In this case, what we're doing is expressing the scaffold so that it has lysines, which would then be a reactive handle to attach a ligand of any design to. For example, we wanted to attach the aptamers or other proteins, but it might be conceivable that we could, in line, just express either if it's a small protein or peptide. For example, actually we've done that in some work that was it's about to be published in Angewon, where we've basically used a similar design, but instead of these lysines, we have the peptide that binds to PA63, the heptamer in Anthrax toxin. We're able to show that we're then able to accurately match the binding, or accurately match the spacing between the ligands in that scaffold to the target and achieve good inhibition there. So
that's very interesting because I was thinking holding of all of them together, like the scaffold holding into the light.
Yeah. I'm not sure that would work, but it might actually also work for the hemagglutinin binding proteins because they are small. They're only about 10 kilodalton, and they do show very good reversible binding. You can measure them, and then they refold very well. So those might also be amenable to that kind of strategy. So it's a good idea.
Thanks a lot. Thanks.
Any other questions? Okay, let's thank Jake again.